Thank you, James. Thanks so much to James and Natalie for hosting the seminar series. And you can hear me okay, right?
Yeah, we're good.
For hosting the seminar series and yeah, for the introduction. Thanks to everyone else for coming to hear about my research today. So the project that I'm going to talk to you about is just one project that I've been working on during my postdoc at the Koch Institute at MIT. And so it involves the chemical tuning of biopolymer fibers from these extensible hyaluronic acid networks. So there are a lot of different reasons that motivate our work in the area of polymer fibers. There are a lot of different applications of this class of materials. We see fibers in our everyday lives, for example, of course, in textiles. They're used in composite materials for enhanced mechanical properties, and as well as of particular interest to the Langer Lab, our tissue engineering and drug delivery applications. And so you can just see one example of sort of a fiber-based biological scaffold here. So in this area, there are different reasons why polymer fibers are really privileged in their use in biological scaffolds, and a variety of different properties that we want to develop and control for in designing such scaffolds. So some of the properties that are really important include the ability to promote things like cell infiltration, diffusion, and mass transport. This is in large part allowed because of the porosity of structures that are formed by fibrous materials, and the fiber diameter is really important in controlling these types of phenomena. So really it enables the diffusion and transport of things like nutrients and oxygen. A fiber alignment is also really important in these structures because it can provide spatial orientation for things like cell growth. And so you can see, for example, in this cartoon here. There are a variety of chemical properties that we want to control for when synthesizing these types of materials and structures. And one really important one is the hydrophilicity of the fibers. This, along with other chemical properties, will affect things like adhesion and the biocompatibility of the material. And then there are a lot of different mechanical properties that we want to control for in preparing these structures. Some examples are, for example, the stiffness of the fiber and stress relaxation properties. And we know from great work from a lot of researchers that these mechanical properties will affect a lot of different biological responses, including cell differentiation. Okay, so in looking at, all of the important applications of polymer fibers, especially in biomedical applications, we saw that there was opportunity to build on existing fiber production methods. And so a lot of these currently used methods that are really common include things like electrospinning, jet spinning, and thermal drawing. And in biomedical research, I think electrospinning is really popular. So there's a little cartoon of what that looks like here. In comparison to these methods, we wanted to develop a method with a few different criteria that we thought would enhance the biocompatibility of not only the method, but potentially the resulting fibers. And so these include developing a process that is low energy, so something that doesn't require high temperature or high voltage. We also wanted to develop something that could work in the absence of organic solvents. The reason being that residual organic solvents can be toxic, right? And then thirdly, we wanted to develop a process where we could achieve desirable mechanical properties using hydrophilic polymers. So oftentimes in some of these processes, we need to use hydrophilic polymers to get the mechanical properties we want, and then go through some sort of reoptimization or coating process to make the materials more biocompatible after the fact. So these were some of the criteria that we wanted to really look at in developing a complementary production method for bioinspired fibers. Right, so what better way to develop bioinspired material than to look at how nature has already been able to achieve these things. So here I'm just showing that, despite a lot of great progress, excellent work, and amazing materials that we're able to synthesize in industry or in the lab, we're still unable in many ways to replicate properties that nature provides, and spider dragline silks are a really great example of this. So when looking at how nature is able to achieve this, there's a few things that we can learn from the natural silk spinning process. One really important thing to note is that the spinning process is known to really control mechanical properties, possibly just as much as the chemical, has a huge influence, just as the chemical makeup of, the starting material has. The spinning process is actually really important in imparting those mechanical properties. The second thing that we know that we want to take home here from, in terms of inspiration from these natural silk spinning processes, is that studies of silkworm silks and spider silks have suggested that the pulling forces, so these forces that are exerted by the animal's body weight or the head of the silkworm, are actually seem to be more important than the shear forces that we observe in the spinning duct. So that's really interesting. It would suggest that potentially developing a process that utilizes these forces would provide enhanced biomimetic properties. And so this is something that we have looked at just. So this is something that we have developed, and in this video, I'm showing just one example of how we can draw out our polymer fibers using this Pultrusion-based method that we've developed in the lab. So sitting on this little piece of paper is basically this fiber precursor, which is a viscous fluid. And we can gradually, like manually or using a syringe pump, draw out these macroscopic polymer fibers. And so what's interesting here is that, similar to that spider silk synthesis, these biopolymer fibers are produced by this pultrusion rather than an extrusion method. And in comparison to, for example, things like self-assembly, we're able to easily access these these fibers that can be drawn. So far, we've seen over 10 meters in length. And so this fiber precursor is actually comes from a reversibly cross-linked polymer network, in this case hyaluronic acid, and only requires water as a solvent. And in this specific project, we focused on hyaluronic acid because of its prevalence in biomaterials, of course. It's biocompatible and biodegradable, but we have also been able to look at some other polymers as well. So some microscopy studies allow us to learn a little bit more about the structure of our materials, as well as give us some insights into some potential applications, right? So SEM shows that we indeed form these smooth cylindrical fibers. And then what I'm showing here in this image is the result of incorporating fluorescent microparticles directly into our fiber precursor, so in that viscous fluid. So basically, in a vial of this viscous fluid containing this dynamic network, can directly incorporate things like microparticles, and then draw out polymer fibers with these fluorescent microparticles dispersed throughout the material. And this is really interesting because it suggests like what we can do with potential drug delivery applications, and also shows how even embedding things like microparticles into our starting material is really orthogonal to this pultrusion process, right? It hasn't really affected the propensity for fiber formation, and it doesn't have a dramatic effect on the diameter of the fiber either. Okay, and then we use polarized light microscopy to learn again a little bit about the internal structure of our materials. And very simply here, this angular dependence of birefringence suggests to us that as we draw this polymer fiber out, we get some anisotropic orientation of polymers along the fiber axis. And then in this image, I'm just showing that our fibers can you can see that they've, to an extent, self-healed or like joined at this intersection in a humid chamber heated to 37 degrees. Okay, so before going into the chemical makeup and characterization of the polymer fibers, I want to show one example of an interesting property that I think arises from the process itself. So as I just said, as we draw the polymer fiber, we get alignment of these polymers along the fiber axis, right? And this dries out and forms this dry, stable fiber. But when we reintroduce water into the system via humidity, we start to observe potentially that this humidity is disrupting the interactions in our structure and leading to an entropically induced supercontraction of our fiber. So it's a little bit hard to see, but this is the sort of starting image where we're starting about 4 centimeter fiber. And then after 15 minutes in a humid chamber, our fiber actually contracts to about 1/4 of its length. And this is a pretty rare phenomenon that is known in spider silks, but pretty uncommon in synthetic materials. And we're also able to apply this property towards humidity-induced weightlifting. So in this direction, we're able to draw up a small weight around 1 centimeter over the course of a few minutes under humid conditions. And I'm just going to show this, just a cute little time-lapse video of what this can look like. And essentially, in this humid chamber, the small weight can be lifted. And we'd really like to look at this over repeated cycles sort of in the next stages of the project and potentially use these contractile properties for muscle mimetic applications. Okay, so now we're going to dive into the chemical makeup and the synthesis of these fibers and the characterization of the fiber precursor, so the starting material fluid, and then the resulting fibers. And it was really important to me to look at the effect of different molecular structure on the properties of those two things, the fluid and the fiber, so that we could understand a bit more about how to rationally design these types of materials. As James mentioned, I am trained as an organic chemist. And so as a chemist, I was particularly interested in how different chemical substituents on our polymer structures will affect the bulk properties of the material. So our fiber precursor in this case is a dynamic network that's made from hyaluronic acid mixed with these boronic acid terminated PEG polymers. And we propose that it's this type of dynamic cross-link that is sort of taking play in this system. Okay, so I was really interested in how different modifications, chemical modifications near the site of cross-linking could affect the propensity for fiber formation. Basically, like, when can we make a fluid that wants to draw fibers, and when do we make a fluid that doesn't want to draw fibers? And then how these chemical substituents would affect the mechanical properties of the solid fibers. So we synthesized a small library of PEGs where we've incorporated, this is an unfunctionalized one, and then different electron withdrawing and electron donating groups on near the boronic acids. Okay, so in order to study to characterize the fiber precursors and learn a little bit more about, how these fibers form, we used dynamic rheology to look at those fluids. And so specifically, we took every one of those PEG polymers in the PEG library that's sort of chemically distinct and mixed them with hyaluronic acid to get a small library of these fiber precursors. For each one of these fiber precursors, we conducted a few key experiments, and here I'm showing a frequency sweep experiment from our Rheology. For each one of these frequency sweep experiments, we see at 25 degrees Celsius a crossover frequency where our storage and loss modulus curves intersect. And we found that regardless of those chemical substituents, these specific crossover frequencies were quite similar to one another, right? Kind of difficult to compare against each other. So we next wanted to look at the temperature dependence of those crossover frequencies. So in this graph, this is an overlay of several experiments on one material. So this is just the fiber precursor that results from mixing hyaluronic acid with the unfunctionalized phenylboronic acid. So one of those materials. So for one of those materials, and for each one of the materials, we ran frequency sweeps at different temperatures to look at the temperature dependence. And you can see that as you lower the temperature of the experiment, your crossover frequency drops and drops and drops. And for each one of these crossover frequencies, we can extract a relaxation time that is associated with large-scale motion in the polymer structure. And so what's interesting is that, when you look at these temperature dependencies, they follow Arrhenius behavior, right? They exhibit Arrhenius behavior. And so you get an Arrhenius plot for each one of these chemical substituents. And we found that the slopes of these plots and the activation energies that we can extract from them actually seem to correlate pretty well with the expected electronic effects of our chemical substituents at the ends of our PEG polymers, right? So what's important to note here is that oftentimes when we think about chemical cross-linking with boronic acids or dynamic crosslinks in general, we often think about what's going to increase the likelihood of cross-linking and what's going to make more crosslinks and what's going to make a stronger network. But in the case here where we're trying to draw out a polymer fiber, right, from a fluid, you can imagine that we would want something that's a really highly dynamic system, right? Something where bonds can form and break really easily. And so I think we see some evidence for that here. From these Arrhenius plots, we're able to extract these activation energies of cross-linked dissociation. And so, as I said, they sort of trend expectedly with our different electronic effects of the chemical substituents. And what we find is that overall, materials that have a lower activation energy of cross-linked dissociation, for those materials, we're able to access longer draw lengths, right, in comparison to those with higher activation energies of cross-linked dissociation. So this, I think, was a very important study that gives us more information about what factors go into making a material that has a high propensity for fiber formation using a pultrusion-based method. And then lastly, I just want to show, of course, some of the characterization that we did in terms of the mechanical properties of the fiber material. For this, so far, we have looked at single fiber tensile testing. And so in the next stages, we really want to build materials in higher dimensions. So right now we've looked at just the tensile testing of single fibers, and specifically how a couple of design handles will affect the tuning of those mechanical properties. The first one here is those chemical substituents that we've been talking about. So what we find with these seemingly subtle changes in the structure of one of our polymers in our system, we're able to observe, in some cases, dramatic changes in the mechanical properties of the solid fiber. So here you can see that with these differences, we're able to observe, sweep out a wide range of stress strain profiles. And I want to draw your attention particularly to the Young's modulus, where, with these subtle changes to the PEG polymer, we can observe really different elastic modulus regimes for each one of our fibers. And so something interesting here is that the methyl-containing crosslinker, which actually had the highest activation energy of dissociation from our previous studies from Rheology, actually produced fibers with really high tensile strength and modulus. And then lastly, this is a really simple way to tune the properties of these fibers that we wanted to look at, which is simply mixing our two polymers in different ratios, right? So this doesn't require any synthetic labor, and you can see some different mechanical properties in this way. So by simply mixing those in different ratios, again, we can see really different stress strain profiles for these fibers, and then also a range of tensile strength and the elastic modulus. And so essentially what we're doing here is affecting the concentration, relative concentration of the boronic acid groups on our PEG polymer. And you can tune the mechanical properties in this way. The reason I've stressed this a lot is just because, very simply, you know, at a high level, you can imagine that using these types of fibers for applications like I discussed in the beginning, you want to be able to tune the properties of these materials because mimicking something like the properties of bone, for example, would be very different from healing something like a tendon. And then I also mentioned previously, you know, in these fiber scaffolds, we've seen that there's a lot of their mechanical properties and viscoelastic behavior causes different biological responses, for example, things like self-differentiation. All right, and so I think my time is pretty much up, but I want to thank a lot of the people who worked with me on this project. It required a lot of, you know, different types of researchers with really different scientific backgrounds. And so I want to thank Bob and Dan, who are my two advisors right now in the Koch Institute, and especially the co-authors on this project. The second author on this project is actually an undergraduate researcher who's been working with me at MIT since the beginning of both his undergrad and my postdoc. And so it's really been a lot of fun working with these people on this on a really multidisciplinary project.
Thank you, Crystal. That was fantastic. So if you have any questions, please post them in the chat, and if we have time, we'll ask them. So we have a few already asked. So Saswat asks about the self-healing properties. So how did you measure that? And can you go into a little bit more detail about the mechanism there of the self-healing?
Mm-hmm. So I would say that's pretty in its infancy. So essentially we haven't measured, what we haven't looked at is, you know, mechanical testing after this healing. So that's definitely something that we'd want to look at more. We just know that, you know, essentially in humid conditions, we can see the fibers sort of join at those intersections. And the reason that we expected that and why we tried that is because these fibers come from this dynamic network, and also because of, you know, the mechanism by which the fibers form. We would expect that basically when they're re-exposed to some water, that you know, those interactions can move around again, and that we would see some adhesion.
Yeah, makes sense. Thank you. So we have a question from Joanna. It says, thank you for the talk. So at the beginning, you mentioned that you tried to use this technique for other polymers. So can you go into that in a bit more detail? Which polymers did you use, and did you see the same type of results? For example, the supercontraction. Did you see that with other polymers? Okay,
so we trying to think of the difference. We have looked at alginate, and we've also looked at polyvinyl alcohol. And we have only done this type of characterization for our hyaluronic acid fibers at this stage. And so I don't believe we've looked at the contraction with the alginate fibers, but based on our proposed mechanism, I guess I would expect that we would see it in that case as well. But definitely something to look into more.
Yeah, sure. Thank you. So questions rolling in. So Simona says, does the temperature above 25 degrees Celsius affect the fiber structure? Or generally, how does temperature affect the formation of the fibers, maybe?
Okay, so in terms of I'll start with, we haven't looked at how temperature affects for example, the mechanical properties of the fibers. Like sort of after they're formed. In terms of the formation, that's a really interesting question that we also haven't controlled for. So everything we've just kind of done at ambient temperature. And oftentimes, you know, in terms of the fabrication, ambient like conditions, humidity, everything like that. So controlling for both of those things, humidity, temperature, those will go hand in hand, of course, moving above room temperature. And I think that the removal of water from our system is like super important in what's going on in there and the resulting properties. So I think it would have a big effect. It's not something we've controlled for yet.
Yeah, I agree. I think one quick question from Tommy. So they say, this is really interesting work. So have you ever considered mixing two different boronic acid polymers to tune the strength or the modulus of the fibers?
Yeah, that's a great suggestion. It's definitely something I want to look at, looking at like mixed types of crosslinks. And I think that would be a really cool way to also look at potentially like dope things in and get gradients of properties right within a material.